Shalini K S from Maharani Science College for Women, Bangalore.
Yeah.
I'll be present. Is my slide visible, sir? Sorry.
Visible.
Yeah. My work is on microwave assisted green synthesis of metal incorporated aluminophosphate composites. And we have studied the photocatalytic effect of these materials in the dye degradation. Now, just to give a background, usually zeolites correspond to silicates, sodium silicates, where the metals are trapped in that. Silica is being replaced by aluminum, which is a trivalent, and phosphorus, which is a pentavalent ion. And then we tried incorporating nickel into or zinc into the material. And the current work I'm presenting is the incorporation of nickel oxide onto aluminophosphate zeolite, so that we have a stable and effective catalyst for photodegradation of methylene blue, which is used as a model pollutant. Now, the scheme of the work is we synthesized aluminophosphate zeolites by microwave method, and then the aluminophosphate zeolites which we synthesized was mixed with nickel nitrate and aqueous extract of Parnelia perlata, which is a lichen which is found in the western Ghats of our Karnataka. In the extract, it is a lichen, so whole plant extract was used as a source for conversion of nickel nitrate into nickel oxide. Characterization studies were carried out by XRD, SEM and EDAX and TEM. We also did surface analysis by BET, and TGA thermogravimetric analysis was also done. The XRD of the nickel oxide aluminophosphate composite is as given here. Now, X-ray diffraction pattern, it confirms that there is orthorhombic phase of aluminophosphate tridimate.
Dr. Shalini, sorry for interruption. Please switch on your video during the talk.
Yes, sir. Okay. Is that fine, sir?
Yeah, you can carry on.
Okay. Yes, sir. So the XRD shows that there is both the peaks corresponding to aluminophosphates and nickel oxide. Nickel oxide. These are the peaks which corresponds to nickel oxide, and this is a peak which confirms the presence of aluminophosphate moiety in the complex that we prepared. FTIR spectra of the nickel oxide aluminophosphate composite is given here. So there is a stretch around 1101 or up to 1106, which is corresponding to metal hydroxide stretch, and the stretch at 725 corresponds to phosphate symmetric stretch, and 539 and 457 are the bending 539 correspond to alumina tetrahedra, and this 457 corresponds to the bending vibrations of phosphate group. The SEM and EDAX are done to analyze the morphology, surface morphology of the composite. As we can see, this is a SEM image of the composite. Average particle size is somewhere around 25 to 45 nanometers. It's agglomerated particles we got. And the EDAX confirms the presence of nickel, aluminum, phosphorus, and oxygen.
Slide is not moving.
Sir?
Slide is not moving.
Okay, sir. This one is fine.
Please continue. Okay, please continue.
Yes. So this is a TEM image of the nickel oxide and aluminophosphate composites, where we can see that the particle size is somewhere around 25 to 45. And this shows that the agglomeration is not as, there are voids that we can see which can act as a catalytic site. BET analysis was done to analyze the surface area and pore diameter and all that. So surface area from BET analysis, we found that it is 49.35 meters square per gram. Pore volume was found to be 0.105 cm cube per gram, and pore diameter was 4.16 nanometers. TGA analysis shows that there is no significant loss of mass anywhere during the course of the TGA study. So it indicates that there is no occluded organic moieties or water molecules in the system. And when we did degradation studies, we did the batch studies for series of studies with varying pH, varying initial dye concentration, and varying catalytic amount, initial catalytic dosage and all that. Now the pH effect was studied first to make sure which pH is more effective in dye degradation, and we could see that under acidic condition in general it was doing better, but normal at neutral pH of 7 we got the better photodegradation effects. Alkaline conditions we did not get much degradation. Actually it was not even giving any degradation. Now when we did the study for catalytic dosage, as the dosage of catalyst increased, the degradation was better, but then if we increase beyond certain age, we got highest percentage of degradation around 8 mg. To make and beyond that 10 mg it decreased. We reasoned it out to be because of the agglomeration of the catalyst in the system. So that closes the active sites and interaction comes down, the photodegradation will be lesser. Now, initial effect of initial dye concentration, when we did the batch studies, we prepared initial dye concentration was stock solution was 50 ppm, and then we took 2 ml, 3 ml, 4 ml, and 5 ml and made it up to 100 ml so that we get a series of 1 ppm, 2 ppm, 3 ppm, 4 ppm, and 5 ppm solution. And then we did the studies to check the initial effect of initial dye concentration on photodegradation. As we can see, within 10 minutes, we got whatever is the highest degradation percentage, we get it before 10 to 20 minutes. That is the initial period, which is more effective as photodegradation, and then it stops. That's probably because all the active sites will get blocked by then, and then afterwards it will be constant. And we got better result at lower concentrations, and as the concentration of the dye increases, the percentage of degradation decreases. And when we did the kinetic study, we could see that the reaction follows pseudo second order kinetics. To sum up the studies, we were able to synthesize the nickel oxide aluminophosphate composites by a facile environment friendly method, and then these synthesized molecules had orthorhombic phase for the AlPO and cubic phase for the nickel oxide. And the surface particle size, average particle size was found to be 25 to 40 nanometers, and 49.35 meters per gram was the surface area. And the molecule was very stable when we did the thermogravimetric analysis. And when it comes to photodegradation at pH 7, we got better degradation of around up to 75 to 80%. The catalytic dosage would initially increase the photodegradation, but later it decreases because of agglomeration. And it follows second order kinetics. Thank you.
Okay. So I was talking about the dyes, which pose threats to the plants and aquatic organisms simply because it is mainly coming from the textile industries or leather industries or tanning industry or something like that. There are many dyes like azo dyes are there, and xanthines are there, then so many such kind of dyes as well as pigments, which cause a lot of threat to the living organisms in the aquatic environment. And because of this, we face a lot of difficulties in treatment of water, especially from the textile industries. So there are many methods also, like physical methods, chemical and biological methods, such as adsorption, ion exchange processes, and advanced oxidation process, and microorganisms and enzymes. All those things have been used for the purification of purification of water, which is polluted by the dyes. Here, we choose a method that is adsorption, which is not causing any harmful effect to the aquatic organisms by creating any kind of residues after the treatment process, and it can be having a recyclability process. Also, it is a cost-effective method and eco-friendly also, as well as it is seen from the literature. So here the main objective of the work is that we have to identify a material that can cause a low-cost material, production of low-cost material, and it must be sustainable and it must be reusable for multiple applications or multiple uses. So next, after selection of the material, what next is the fabrication of the material, whether it should be in the form of powder or it should be in the form of film or in the form of bead. So that has to be the next selection. And afterwards, where we are going to apply it, it must be applied for the adsorption process, or desorption has to be carried out. Again, reusability of the material has to be done. So this is the main objective of the process. Whereas when we go to the methodology, what we have identified is that India is creating a lot of agricultural waste. It is not a creation, as we know that it is a part of the food that we are consuming daily life, like the peels of banana and the pomegranate peels or orange peels or watermelon rind. All these things are available in large quantities. So we have selected certain peels, fruit peels, and it has been powdered and it has been used for the adsorption process. So coming to the preparation of the adsorbent, how we have done this material or produced this material, we have collected different kinds of peels and it is dried for the removal of water content from this, and it is powdered. The sieving process is done to get the fine powders, as we know that when the particle size decreases, it can provide more surface area for the adsorption process. And then it is studied for the adsorption of particular dyes like crystal violet and methyl orange from the aquatic environment. Key findings, we have gone for only the FSM analysis of the material to see the structural morphological characteristics of the material, so that we could find that the material of orange peel, pomegranate peel, watermelon rind, and banana peel have been shown granular nature, which is having irregular shape. It is due to the traditional grinding process and the sieving process that we got this kind of mixed images. And we have gone for the adsorption studies, as I have already indicated, for the methyl orange as well as crystal violet. Different parameters have been studied. One among the parameters is initial dye concentration. As the dye concentration increases from 2 to 10 ppm, the adsorption rate also increases in the case of almost all the 4 materials, in the case of crystal violet adsorption as well as methyl orange adsorption. So this is due to the presence of more active surface area for the adsorption of dye molecules on the surface of the adsorbent. While talking about the effect of time, as the time increases, adsorption rate also increases. But at the time of around 300 minutes, that means maybe around, or 360 minutes, maybe around 6 hours, end of the 6 hours, it becomes saturated adsorption process. Why? Because almost all the active sites which is ready for the supply of the sites for the adsorption process has been completely saturated by the end of 6 hours. So it's a slow adsorption process, and it has been completely adsorbed afterwards. There is no adsorption has been observed, so that it is in the equilibrium state. And we have gone for the kinetics of adsorption to check whether it is pseudo first order reaction or pseudo second order reaction or intraparticle diffusion model, whatever the type of adsorption that we can go through. And we have analyzed for the both CV as well as methyl orange adsorption studies and found that it is following pseudo first order adsorption model, with which is determined from the regression coefficient value, because pseudo first order regression coefficient value is almost near to 1 compared to that of the second order one, and the maximum dye adsorption values have been obtained with almost 0.68 for banana peel, and for orange peel around 0.8 milligram per gram, and pomegranate peel it is around 0.4 nearly milligram per gram, and watermelon rind around 0.7 milligram per gram of crystal violet from a solution of 50 ppm concentration. Similarly, even in the case of methyl orange also, we have obtained almost a similar kind of result. It says that the adsorption process depends on diffusion of adsorbate into the surface of the adsorbent molecules, and hence it is proved that it is a physisorption process. Again, in order to confirm the presence of the formation of physisorption or the occurrence of physisorption process, we have gone for isotherm models, which we have applied was Freundlich's adsorption isotherm model as well as Langmuir adsorption isotherm model. And we found that based on the regression coefficient and based on the degree of linearity of and based on the extent of adsorption, we found that the Freundlich model is the best fit for the adsorption of crystal violet as well as methyl orange from the aqueous media. And it indicates that Freundlich model always indicates that it is a reversible process as well as multi-layer formation. So obviously there should be a multi-layer formation because we have used the fruit peel extract, and which will contain a lot of chemicals in that. So there must not be any kind of particular or specific material that is responsible for absorbing the anionic or cationic dye on the surface of this kind of materials. So what we have observed is there is a multi-layer formation of the adsorbate molecules on the surface of this fruit peel waste. That is why it is obeying the Freundlich adsorption isotope model. Similarly, different temperature has been employed for the study of this adsorption process, and found that it is a non-spontaneous process which is indicated by the positive value of the Gibbs free energy, change in Gibbs free energy. And similarly, the positive value of delta H value, enthalpy value, indicates that it is an endothermic process. So adsorption makes use of some kind of energy for the process. So as the temperature increases, there is no much change in the adsorption process. So since there is a need of temperature for the adsorption also. And similarly, the positive value of delta S is also indicating that there is a random movement of adsorbate molecules on the surface of adsorbent, and this random movement could be due to the Freundlich adsorption isotope model. We can explain with the support of Freundlich adsorption isotope model that one way the first layer of molecules are getting adsorbed on the surface of this adsorbent, and again second layer is getting adsorbed on it, and maybe multi-layers are forming. Afterwards, after some time, maybe as I told in the previous case, that maybe after 6 hours, there may be a desorption process also. So we found that 6 hours will be the optimum time condition that can be used for the adsorption of any of the dyes on this surface of this material. To conclude from my work is that fruit peel have been used as adsorbent very successfully, and the efficiency of the removal increases with a higher dosage of the adsorbent. Similarly, it obeys pseudo first order kinetic model, and it follows Freundlich isotope model as well. And the different adsorption capacities, as I have already mentioned in the previous slides, and the removal efficiency of each adsorbate for both crystal violet as well as methyl orange was found to be almost 90% in 6 hours of time. And this is the preliminary study of our work, and the future plan will be focusing on the chemical modification of the hydrogel bead. That means we are going for the production of the hydrogel bead with this fruit peel waste, which is going to be a challenging work for us because there are no industries, textile industries, which are working with these kind of hydrogel beads for the purification of textile water. And it will be tested with model solutions, as I have told, that aqueous solutions of methyl orange or crystal violet or different dyes, which have been mainly used in the textile industries. Then we will go for the scale-up studies because we are closely related to our other sister industries like Sriram or Scotta, something like that. So we'll be going with the scale-up studies. So we have started with the preliminary studies, and we'll be going for this scale-up studies in coming future also. And thank you very much. I'm open to have any kind of queries from your side, and you can contact me through my email address or contact number. Thank you. Thank you very much.
Okay, ma'am. Thank you for giving me an opportunity to present a talk on this session. This is Dr. Nookaraju, Associate Professor of Chemistry, Aditya College of Engineering and Technology, Sarumpalam. Today, I would like to present a brief talk on importance of functional MCM41 materials in synthesizing the pyridine 2 carbaldehyde derivatives under functionalized MCM41 materials. So my talk is going to have importance of introduction towards the MCM41 materials. As you know, the materials which are porous in nature have been classified into 3 different categories, such as microporous, mesoporous, and macroporous. Among these materials, mesoporous materials, which are having a pore diameter of about 2 to 50 nanometers, are found to have well advanced characteristics of functionalization on the surface, which can have the adsorption of large organic molecules, dyes, and the chromatographic separations. That's why we have chosen a material which is a silica-based MCM41 material to have the catalytic application, which we can say is a reusable catalyst. And the MCM41 materials are going to have the hexagonal pore structure, and functionalization of this Is my slides visible? Hello?
So,
Visible but not
Repeat.
moving.
Not moving?
if it is not
Yeah.
moving,
Yes, right. Now is it mover?
Yes, it is moving.
Yeah. So these mesoporous MCM41 materials have been widely used as a heterogeneous catalyst, and they are going to have a surface OH groups where we can functionalize them with the metal ions as well as the acidic groups, so that we can use them for the synthesis of fine chemicals and very important pharmaceutical compounds. So our objective is to develop a one-part multi-component synthesis using functionalized MCM41 materials, which will be an eco-friendly approach. So the chalcones, which are well known as alpha-beta unsaturated ketones, have very important viable pharmacological applications such as antifungal, antimalarial, antioxidant, and anti-cancer activities. So here we have synthesized MCM41 materials by a room temperature co-precipitation method using a surfactant CTAB, and which we have used by using a room temperature method. So above this MCM41 materials have been synthesized by a room temperature method by using CTAB as a surfactant. And these materials have been functionalized with various metals such as titanium, cobalt, manganese, aluminum, etc. And also functionalized with the sulfonic acid and phosphotonistic acid in order to enhance the acidic character at the surface. So these materials have been characterized for their mesoporosity as well as the crystallinity and their surface characteristics by various sophisticated analytical techniques such as X-ray diffractors, nitrogen adsorption desorption isotherms by BET model and scanning electron microscope, and also by DRS UV visible, so that we can know the metal content that has been present in the material. And after confirming the mesoporous character of these materials, we have moved to the catalytic studies in the synthesis of chalcone derivatives at under solvent-free conditions. And for this, we have taken acetophenone and pyridine to carbaldehyde, and we have added 1 millimole of mesoporous MCM41 materials, and this was reflexive for about 1 hour to have a solvent-free condition. And all these materials, the derivatives that were synthesized, have been characterized by their IR and NMR, which will give a clear information about the functional groups that have been incorporated during the synthesis process. And this is the plausible mechanism for the synthesis of pyridine 2 carbaldehyde derivatives, which are chalcone derivatives under solvent-free conditions. The advantage of this methodology is eco-friendly, solvent-free room temperature, low temperature synthesis, which is a main objective of green chemistry. And these are the some what we say reactants in the products as per the criteria. And whenever we come to the catalytic efficiency of these materials, among the materials that was tested, phosphotonistic acid MCM41, which is having a more acidic functionality at the surface, is found to have an yield of about 92% in compared with the unfunctionalized MCM41. And we have studied the antibacterial activity and antifungal activity of these derivatives that were synthesized by the MCM41 materials, and we found good results in agreement with the antibacterial activity as well as the antifungal activity. These are the results that shows all the derivatives like 3A to 3E, that means 5 derivatives we have synthesized by using this, and all this have been presented over here. And in order to conclude, we have a one-part synthesis route for the chalcone derivatives under solvent-free conditions, and which is a unique in its form. And these materials are highly efficient in bringing up chalcone derivatives on high yields under normal experimental conditions. And this is a green route, a very effective protocol in order to have the synthesis. And among these, phosphosolic acid material is more effective for the catalytic conversion under room temperature condition, under solvent conditions. So, in order to acknowledge, I would like to thank the organizing committee, the research platform publishers, as well as the CRSI, Bihar and Jharkhand local chapter, and more importantly, GB College, Ramghav Department of Chemistry for their organization. And I would like, last but not least, I would like to thank our principal and the management of our college for their support. Thank you.
Myself, Monika Joshi from Amity. The title of my talk is Nature-Inspired Versatile Superhydrophobic Nanocomposite for Oily Wastewater Remediation. I've tried to align my talk with the theme of the conference. So this is the basic, the outline of my talk, motivation part, introduction, by conclusion. As we know that for a good environment, we need a safe drinking water, air, good air, and good food, good quality of food. But unfortunately,
Dr. Monika, you
Yes,
are
sir.
not.
yes, sir. Yes, sir. Hello.
Please continue. Is there any
Okay.
problem?
Yes, sir.
Your voice is not clearly audible. That's it.
Okay, sir. Now, so for the good water, for a good health, we need a good drinking water, good food, good air. But unfortunately, all these fundamental needs for human beings are missed nowadays. Our air, as well as our water, even our food quality is contaminated by variety of contaminants. So today, I'm discussing here the sources of oily wastewater and the removal techniques, the elimination of separation of oily wastewater from the using nanomaterials. So coming to the talk, we know that, as we know that there are the variety of sources that generates wastewater. Like the one can expect that only the domestic discharge is the source of this wastewater, but apart from that, our industrial wastewater that is contaminate our all the water bodies, like our food industry, textile industry, automobile industry, oil industry, pharmaceutical industries, and so many other industries give a lot of contaminated water. So we have tried to check the presence of the oil droplets in oil industry wastewater. Basically, that water we have collected from oil industry, that after treatment we have collected it and chemical parameter. You can see the two second row and third row, second column and third column, and these are the Center Pollution Control Board guidelines for the discharge of permissible limits for the discharging of the wastewater, and but.
So, namaskar to all the respected dignitaries and dear participants. I, Dr. Rakhi Khandelwal, Assistant Professor, Government Mahila Engineering College, Ajmer, Rajasthan, now presenting my paper on green synthesis of silver nanoparticles, eco-friendly method and versatile application. My paper ID is 091 and I have a total number of slides 15. So, here is the outline of my presentation, which includes introduction, materials and method, characterization, applications, and conclusion. So when we think about the title of my talk, that is green synthesis of silver nanoparticles by using plant extract. First, the question comes to our mind that why silver nanoparticles, why green synthesis, and why we are choosing the plant extract for this synthesis method. So let us begin with the first question, that why we have chosen silver nanoparticles for our study. We know that silver nanoparticles are nanoscale particles of silver, typically ranging from 1 to 100 nanometers in size. And due to the various unique properties of silver nanoparticles, they are of very practical uses. Such properties are size-dependent properties such as optical, electronic, and catalytic properties, which are making the silver nanoparticles for versatile applications. Second one is the antimicrobial activities. The silver nanoparticles possesses potential antimicrobial properties against a broad spectrum of various type of bacteria, fungi, and viruses. Third one is the optical properties. The silver nanoparticles exhibit strong absorption and scattering of light.
Dr. Rakhi, your slides are not moving. Please click on the slide so that it can move.
Sir, I am in my view, all the slides are moving, sir. I have now on slide number 4.
It is.
Sir, I have now on slide number
My
4.
screen, it is not moving. No, my screen is showing only slide number 2.
Okay, sir. So I skip from the slide
Now
show.
it's moving. Now it's moving. Now it's slide number 4, introduction.
Okay, sir. So actually, I think due to animation, this not work.
Yeah, you have to click on each slide so that it can display.
Okay, sir.
Yeah.
Okay, sir.
Now it's fine.
So now, second question is the why green synthesis? Basically, we know that the silver nanoparticles can be synthesized by broadly two types of method. One is the traditional or chemical synthesis. Second one is the green synthesis. As we know that the traditional synthesis method of silver nanoparticles typically involves chemical reduction techniques where silver salts are reduced to nanoparticles using various type of reducing agents, and after that the silver ions get converted into silver atoms, and after that due to the nucleation process the formation of the silver nanoparticles takes place. But by question comes, why green synthesis is better? As we see that chemical synthesis involves use of various type of chemicals such as the silver salt solution, reducing agent, stabilizing agent, solvent, etc. But in case of green synthesis method, all these chemicals are not required. In this synthesis process, we require only the silver salt solution and the plant extract. So when we are using the green synthesis method, these methods are eco-friendly and can be easily manageable, as well as these methods can be carried out at room temperature, and also these are the cost-effective method. Now, third question comes to our mind, that why we have chosen the plant Phaseolus vulgaris. In our study, we have prepared silver nanoparticles by choosing the plant extract of seeds of Phaseolus vulgaris. Why we have chosen this plant? Because this plant is easily available at the nominal cost. It is the edible plant and has no toxic constituents, which make it suitable for the preparation of silver nanoparticles. And after the synthesis, these synthesized silver nanoparticles can be used for the medical application. Thirdly, the phytochemicals present in the extract act as a reducing as well as the capping agent, so no other additional chemical is required for the synthesis. Now, coming towards the experimental part, the materials required in the green synthesis method is only two. One is the extract of seeds of Phaseolus vulgaris, and second one is the silver nitrate solution. To prepare the extract of seeds of Phaseolus vulgaris, we have collected the seeds from the local market and then ground it in by and converted it into the powder of the seeds. After that, this powder is soaked in the doubly distilled water up to 20 hours, and after that we have centrifuged it at 15 minutes at 5000 RPM. So that is how we prepare the extract and the silver nitrate solution. The concentration of silver nitrate solution we have taken 0.01 m. Now coming towards the experimental part, to prepare the silver nanoparticles, we have mixed the plant extract with the silver nitrate solution in the 1:10 ratio. when we mix the plant extract into silver nitrate solution, actually the color changes from transparent to light brown and the reddish brown color, which indicates the formation of silver nanoparticles. And after the preparation, after the color changes, after 24 hours, we centrifuge the solution and converted it into the dry powder of the silver nitrate. Actually, what happens that when we mix the silver nitrate with the plant extract, it converted into the silver ions and nitrate ions. And the biomolecules present in the plant extract, that is phenols and flavonoids, get reduced the silver ions into the silver atoms. Now these silver atoms get nucleated, and with the help of protein present in the plant extract, which are act as a caping agent or stabilizing agent, get converted into the stable silver nanoparticle. So that is how we have prepared the stable silver nanoparticles with the help of plant extract. Now, after the preparation of the silver nanoparticles, we have done the characterization. So first we have done the characterization UV visible spectroscopy. Now UV visible spectroscopy, in the UV visible spectroscopy, we have obtained two peaks, the SPR peak in the range of 450 nm and the additional peak also we have obtained in the range of 250 nanometer, which could be due to the transition in which involving the silver nanoparticles with the higher multipoles. So the silver UV spectra confirms two things. One is that the symmetry is less than the sphere. Second one is the silver nanoparticles formation is confirmed by the UV visible spectroscopy. Now, next we have characterization by the FTIR analysis. Actually, sir, I have incorporated the animation, and due to the not available in slideshow, so that is why I am changing here. Sorry for that.
No problem. You just take care of this.
In FTIR analysis, we have shown the peaks at 3310 to 3364 centimeter inverse, which shows the OH stretching vibration of the hydroxyl group, hydrogen bonded alcohol, N. Phenols or NH stretching of primary and secondary amines and amides. And it also shows the peak at 1651 due to the carbonyl stretching of the primary amides of the proteins. This is close to that reported of the native proteins, which support to the point that proteins are interacting with the green synthesized silver nanoparticles as a caping agent or the stabilizing agent. So the FTIR analysis confirms the involvement of the phytochemicals in the formation of silver nanoparticles. After that, we have performed the XRD measurement for the size confirmation of the silver nanoparticles. The XRD spectra, in the XRD spectra, various peaks are shown at the different degrees, and which corporates to the planes 110, 111, 200, and 211 crystal plane. The XRD spectra shows that the particles are crystalline in nature and cubic in the structure. The particle size calculated from the XRD data is 15.76 nanometer. After that, we have studied the biomedical applications of the synthesized silver nanoparticles. First, we have performed the antimicrobial activities. The antimicrobial activities of synthesized silver nanoparticles was studied by Dix diffusion method against 3 bacterial strains, that is Pseudomonas aeruginosa, Staphylococcus aureus, and Klebsiella pneumonia. Against all the 3 bacterial strains, the synthesized silver nanoparticle shows very prominent, significant result. That means it shows that these synthesized silver nanoparticles effectively inhibit these 3 bacterial growth. Second one is the anti-cancer activities. The MTT, for the anti-cancer activity, we have done the MTT assay. The MTT assay results for synthesized silver nanoparticles against the human breast cancer MCF7 and mammary cancer EVSA7 cell lines are shown in this table. In this table, we can clearly see that the in presence of synthesized silver nanoparticles, there is this significant reduction in the cell number of the cancer cell, tested cancer cell. That means presence of silver nanoparticles reduces the rate of cell proliferation. That means these can inhibit the growth of cancer cells. Now moving towards the conclusion part, this green approach produces silver nanoparticles in a clean, non-toxic, eco-friendly, cost-effective, and efficient manner at room temperature without any additives. The silver nanoparticles were successfully synthesized with the particle size of 15.76 nanometer and the crystal structure is cubic. We are using the green principles here to synthesize the nanoparticles. These green principles are use of less chemicals and use of less toxic chemicals. The silver nanoparticles with the phytochemicals can easily penetrate the bacterial cell and inhibit their growth, hence showing good antimicrobial potential. The phytochemicals helps to improve the susceptibility of the cells of silver nanoparticles, and due to nano size, this increased surface area improves the penetration of silver into the bacterial cell. The silver nanoparticles showed potent cytotoxicity against the tested cancer cell lines MCF7 and EVSA7. These silver nanoparticles were synthesized by eatable plant seeds. Therefore, these are kept with the phytochemicals of these plants, and this is the reason that is why these green silver nanoparticles may prove to be more susceptible for human body, and when targeted on the cancer cell, these can inhibit the growth of the cancer cell. That's all from my side. Thank you so much. I sincerely express my thanks to UGC DAE CSR Indore Center for providing financial assistance for the research work and for providing the UV visible and XRD studies facilities. And I am also thankful to Shri Ram Swarup Memorial University, Lucknow for providing facilities for studies of biomedical applications. I am also very thankful to the chairperson of the session and the organizers of this prestigious conference for providing me the opportunity to join the conference and present my work. So, thank you so much.